These experimental and analytical methods provide guidelines that aim to understand the role of the many components of the central nervous and musculoskeletal systems in human postural control.
Postural control models with meaningful physical parameters can be used to investigate the role and interaction of the sensory systems and their changes due to disease and aging.
These methods can be used to assess patient balance problems, to reveal the etiology of the impairment, and to aid in the design of interventions for improving postural control.
These methods can also be used to study the interactions between sensory-motor pathologies and balance control, for example, for fall prevention in the elderly.
This protocol provides a means to investigate the relative contributions of sensory modalities, including proprioceptive and visual systems, and their interactions as well as muscle passive contributions to postural control.
To prepare a subject for the measurement of electromyography from ankle muscles, use single differential electrodes with an interelectrode distance of 1 centimeter. Mark the medial gastrocnemius at the most prominent bulge of the muscle, the lateral gastrocnemius at 1/3 of the line between the head of the fibula and the heel, the soleus at 2/3 of the line between the medial condyles of the femur and the medial malleolus, and the tibialis anterior at 1/3 of the line between the tip of the fibula and the tip of the medial malleolus. When all of the points have been marked, use a razor to shave each area.